Good morning, everyone. You are now about to start Unit 3, which is officially about sickle cell disease, but it's really about genetics. And we're going to be focusing on the genetics part in our abbreviated virtual learning um, session. So just be aware of that. If you've had biology, a lot of this is going to be review of what you've had in biology. So that'll be good. You'll see it in a different way, but it will be very much the same kind of stuff. So this is going to walk you through, this video is going to walk you through lesson 3.1.1 or activity 3.1.1, which is kind of the introduction to sickle cell disease, and still using Anna as our framing device. I'm going to go ahead and share my screen. There's going to be times when I'm going to tell you to pause the video to do some research or work through some steps. So just make sure you do that as you are working on this assignment. So I'm going to go ahead and share the screen. And I am now sharing my entire screen. So I'm going to go over to the PowerPoint, and we're just going to walk through this really quickly. I'm going to show you how to do some of the math that's involved and just some other things like that. This slideshow is going to be available in the resources tab. So if you want to follow along with the slideshow rather than using this video, you can do that. Although some of the things I'm going to say aren't written in the slideshow. So the first thing is the autopsy report. So we're introducing Anna's disease just like we did before with diabetes. So we're going to take a look at our autopsy report. So here's my autopsy report. What we can see is our external examination. The nails on Anna's fingers were blue or bluish gray in color. Take a moment and guess what you think that would mean. And if you guessed that bluish gray fingernails mean that she was getting poor levels of oxygen in her blood, you were right. Now we have our toxicology reports. In her blood, we found traces of hydroxyurea, which is a drug used to treat sickle cell anemia. If you think way back to the crime scene, when we saw Anna at the crime scene, we saw that she had a prescription for hydroxyurea, and I had you look up what hydroxyurea does, and it's used to treat blood cancer, and it's also used to treat sickle cell anemia. How it works is it forces the body to switch to making a different form of hemoglobin. You're going to be learning about the mutation that causes sickle cell disease. It results in the hemoglobin, which is the oxygen-carrying protein in red blood cells, being misshapen, and it doesn't work correctly. Hydroxyurea forces the body to switch to a different form that's not affected by that mutation. So now there's some other things in here that you're going to fill out, either typed or written, depending on which one you'd rather do. You're going to learn about a test called hematocrit, and we're also going to be looking at what's called the microscopic analysis of blood. And then you're going to speculate on possible causes of death. These are going to be causes of death that could affect somebody with sickle cell disease. So now you're going to pause the video in just a moment, and you're going to go ahead back over to the procedure in Canvas, and looking at steps 1 through 6, you're going to research the 4 main components of blood. There's a chart in there with their names, so you just need to look those up, write a sentence or two about what each of them does, and you can either type that chart or you can write it by hand, and then answer conclusion question number 1. So go ahead and pause the video for a moment and do that research. All right, so you've now paused the video. You've completed this chart. You've worked your way through sickle cell disease as a blood disorder. You have these 4 components: plasma, red blood cells, also known as erythrocytes, white blood cells known as leukocytes, and platelets, which are known as thrombocytes. So you have determined the function of each of those things. And now we're going to take a look at normal blood and Anna's blood. So ordinarily we would have done this with a microscope, but since you don't have a microscope at your house, well, maybe you do, I don't know. Instead, we're just going to look at some pictures. So back to our slideshow. What we have right here on the left-hand side, we have normal blood. You can see the red blood cells. the white blood cells and the platelets. These little dots are platelets. And on the right-hand side, you can see a slide of Anna's blood. We have red blood cells, white blood cells, and platelets. So take a moment, look at the difference between those two, and then you can pause the video and go ahead and draw what you see. So as you looked, you could see that this normal blood, it kind of looks like SpaghettiOs. These little red blood cells, they're round and they're a little darker around the edges than they are in the middle. The reason for that is a normal red blood cell is shaped sort of like a Werther's, if you're familiar with those. They're disc shaped and they're a little thicker around the edges than they are in the middle. They're kind of thin in the middle, but they're nice and round. Over here, you can see that Anna's red blood cells are not shaped roundly at all. They look like little bananas or little spindles or little sickles, which a sickle is an old agricultural tool. It's like a big knife and a stick. Not exactly what the Grim Reaper carries, but kind of. So you can see that they're abnormally shaped. They're long and pointy and kind of curved like a little crescent moon. So make sure that you draw that. That's one of the things you have to turn in is your drawings of the normal red blood cells and Anna's red blood cells. So go ahead and pause the video now and draw what you see right here. All right, now you're done drawing. So this is one of those days where there's lots of different things. So you've now seen Anna's blood and normal blood. So the source of most of the problems that sickle cell disease causes is caused by the difference in shape of the red blood cells. Part of it is that they don't carry as much oxygen, and we'll get to that later, but it's also a matter of their shape and the effect of that on the blood vessels. So right here under step 15, what you have is a video that another biomedical science teacher made. You would have done this in person in class, but her demonstration shows the difference pretty well. The red cherry, I guess they're cherry, maybe they're strawberry, I don't know. The red runts represents normal red blood cells. And the mouth and the opening of the flask represents a really, really small blood vessel, a capillary, which are the smallest blood vessels in your body. And those are the blood vessels that actually carry or allow nutrients and oxygen to get out into your tissues. They're so small that red blood cells kind of have to go through them single file. So you can see how the red blood cells flow when they're normally shaped with the red runts. And then she has the yellow banana runts that are going to demonstrate the sickled red blood cells. And you can see what happens when she tries to pour each of those things out of the flask. So go ahead and pause the screencast video and watch this video. And then answer conclusion questions 3 and 4, which are at the bottom of the Canvas page. All right, so you should have seen that the normal round red blood cells flowed easily out of the mouth of the flask. She could pour them out really, really easily. In contrast to that, the sickled banana-shaped red blood cells, they got stuck. They kind of tangled together and clogged up. And that's actually the source of most of the problems that sickle cell disease can cause. Basically, those sickled red blood cells get caught in the very, very tiny capillaries. And depending on what organ that happens in, it can cause pain in the joints. It can cause damage to an organ because remember, any organ that loses its blood supply gets damaged because the tissue starts to die. It can cause a stroke. A stroke is a clog in a blood vessel in the brain. That can be really, really dangerous. It can cause what's called a pulmonary embolism, which is the same thing, a clog in a blood vessel, but in the lungs instead. It can cause a heart attack. It can cause kidney damage. A really common thing that it can cause is a thing called splenic sequestration, which is when the spleen, which is a blood filtering organ, gets clogged up with red blood cells and swells, and then eventually it bursts, and then the person loses a lot of blood and potentially even dies. If you Google how did Anna Garcia die, that's the one that comes up. That's not how she's going to die this semester, but that is one possibility that sickle cell disease can cause. So now, we're going to move on to part 3, which is about anemia. So you may have heard of sickle cell disease referred to as sickle cell anemia. And you can see right here in this paragraph that healthy red blood cells live between 90 and 120 days. Your body removes them. There's a hormone that tells your body to make more. Sickled red blood cells don't live as long as normal ones, which means that the supply of red blood cells is short. In your research on the parts of blood, you learned that the cells in blood, especially the red blood cells, are kind of floating around in the liquid plasma. And it's a suspension, which means you can actually settle the red blood cells to the bottom of the blood to see how the different proportions. The way you do that is when you do that, it is a test called hematocrit. And all the hematocrit is a measurement of what percentage of the blood is made of red blood cells. The way you do that is you take what's called a centrifuge, which is a little machine that, here we go, it looks like this. There's a centrifuge, and each of those little things is a test tube, and you have to put it in at a balance, otherwise it gets like a washing machine that's out of balance and it starts rocking around and then it breaks. But you put your test tubes in here, these little microcentrifuge tubes, you close the lid, you turn it on, and it spins really, really fast, and it settles the red blood cells to the bottom of the column of blood. So here we are back to our presentation. So you take your whole blood, you put it into that centrifuge. The plasma is the least dense, so it stays on top. The red blood cells are the most dense, so they sink to the bottom. On top of the red blood cells, you get a really, really thin layer of white blood cells and platelets called the buffy coat. Kind of just looks like an oil slick on top of the red blood cells. So you do that, and then if you have a nice rectangular tube, which is actually what you use in a hematocrit test, is a really, really skinny rectangular bottomed test tube. You can measure how high the column of red blood cells is, compare that to the height of the entire column of blood, and that gives you a percentage, and that's your hematocrit number. So what we have right here is a hematocrit tube simulated. So what you can see, this is Anna's hematocrit. So this entire column is the height of her blood. I've included a ruler right here, so you don't have to get out your ruler and hold it up to your screen to measure. This right here is the red blood cells. This little thin, darker yellow thing is the white blood cells and the buffy coat. So what you do is you see that the whole height is 8, and this is not any particular unit of measurement. You'd ordinarily, and it doesn't matter what unit of measurement you use because you're just dividing height by height so the units cancel out and go away. So what we have, you can see that her red blood cells are 2.4 units high, and the total column of blood is 8 units high. So if we open up our calculator, we divide 2.4 by 8, and I will let you do that on your own at home to get the final number. So go ahead and plug that number into your calculator and figure out what you get. All right, so you should have gotten your final number. I'm not going to give away the answer. So here's the formula, just in case. Red blood cell level divided by total blood level times 100 equals the percent red blood cell volume, which is equal to hematocrit. It's just a percentage. So to remember that formula, just remember it's the percentage of the blood that's made of red blood cells. So if you look, you'll notice that there's a table right here under step 20 that includes the normal number, the normal range, and the low and high numbers. So take a look at the number that you got and determine whether Anna's number is high, low, or normal. Once you've done that, go ahead and fill in the results on the autopsy report page. Right here under hematocrit, you should write out your math. So 2.4 divided by 8 times 100 equals whatever level, whatever that math comes out to that I let you do yourself. You can use a calculator. And then right next to it, write whether that is high, low, or normal. And if somebody has normal hematocrit, they're just considered normal. If somebody has high hematocrit, we call that polycythemia. And if somebody has low hematocrit, we call it anemia. So low hematocrit is called anemia. Normal is just normal. There's no special name for it. And high is called polycythemia. So now you figured out, does Anna have anemia, normal hematocrit, or polycythemia? And then you can even compare her hematocrit level to the normal to figure out if she's really low, really high, or kind of close to normal. So now what you need to do is you need to make sure you have your autopsy report filled out with your hematocrit results, your microscopic analysis of blood. This is where you'd write out the difference in shape between her red blood cells and what it should be. You can even do your drawing on here if you print this out. And then possible causes of death, you can do some Googling to figure out possible causes of death that could be caused by sickle cell disease. So list off, I'd say 2 or 3. That should be plenty. Now all you need to do is answer the rest of the conclusion questions, and you can turn this in. So as you've watched this video, you are probably pretty much all the way done. Stay home, stay safe, and thanks for watching.